This conference will now be recorded.
Well, since I hope everyone is here and we have a lot to cover, we really should begin today's webinar. I really appreciate you all for attending today's virtual conference day 2. A very good morning, good afternoon, or good evening to one and all present here virtually with me today. Let me introduce myself. My name is Clara McBroom, Conference Secretary at Coalesce Research Group LLC, and I will be available if any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. Finally, I would like to thank all the attendees and participants of today's virtual conference on pharmaceutics and novel drug delivery systems. And I hope you enjoy the webinar and gain the knowledge from the sessions today. We all hope your participation will be contribute to new research findings and that will lead to new collaborations and research initiatives. Before we start with the session, I guess some of you will be new to GoToMeetings app. Let me just explain you the important tools required for the presentation. On the GoToMeetings app or a web page, you can find your icons at the bottom center: mic, camera, screen, end button. Mic button, click on that to mute or unmute yourself. I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera button, you can use this button to share or unshare the web camera. If you have the option to share the web camera, you can do it and it is completely your choice. Screen button, you can use this button to share your screen. This is the main part. During your presentation, after I call your name from my side, I will be making you as a presenter and will give the permission to share your screen. Then only you will able to share your screen. So once I make you as a presenter, you can click on the screen button and you will find a pop-up window to share your screen. End button, this is the fourth icon where you can use this to leave the session. On the top center, you can use the option to change your display settings. At the top right corner, you will have three icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For settings, you can use this button to check for the settings for your speakers or microphone. So as I mentioned, kindly remain muted during the presentation so that the presenter does not get disturbed. And for any questions, you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you to please give us a brief introduction about yourself before starting this session so that we can know you about virtually. Now, let's move into our session. The first presenter is Dr. Noro Jebli from Abdel in Badi Mustaghanam University of Algeria. Hello, Dr. Norendin. Are you ready with your presentation?
Yes, it's okay.
Okay, Dr. Noor, I'm making you as a presenter. Kindly click on the screen button to share your screen.
It's okay.
Yeah, your screen is visible now. Click on the presentation mode and start your presentation.
It's okay.
Yeah, it's perfect. Now you can start your presentation.
Okay, sorry. Okay, good morning, everybody. Did you hear me?
As Doctor, you're audible. Can you check your mic settings as there's a lot of disturbance from your end?
Did you hear me now?
Yeah, your voice is audible, but there's a disturbance from your end so that your voice is not clear.
Oh, sorry. And now?
And it's perfect now. You can start your presentation.
Can I start?
Yeah, you can start. You can start. It's
Okay.
perfect now.
Okay. Uh, Good morning, everybody. First of all, I want to thank Peter Jones for his invitation to give me this opportunity to present my research. Please, I introduce myself. I am Professor Noureddin Jibli from Algeria. Algeria is in the north of Africa, Mediterranean country, and I am chair of Laboratory of Pharmacognosy and EP Phytotherapy in Mostaganem University in the west of Algeria. Okay, my talk about today is about neuroprotection of natural product against neurodegenerative disease. Some information. What happened? To pass. Sorry. To pass in the next. I cannot. Sorry. What happened? Okay. Some introduction or my introduction of my talk is about neurodegenerative disease. If you know that interest research effort of the past 2 decades to understand the pathogenesis of neurodegenerative disease and design effective therapeutic have been focused mainly on neuron. That all to this court and review or contribute to our understanding neural dysfunction did, but this suggests that the pathogenesis of neurodegenerative disease order is more complex than previous two, and that the lack of success of neurogenetic path of rapid made you. That this is different factor associated with neurodegenerative disease, as we have genetic factors, age, and we have environmental factor. About the second factors as the problem of oxidative stress, as aluminium, about Alzheimer's disease, and lead about Parkinson disease. This is the figure present different factor associated with neurodegenerative disease in general. About pathologic protein, for example, about Alzheimer's disease, it's same of amyloid beta, and Parkinson disease as alpha-synuclein. The protein of tau is the same of to this pathologic. Okay, now what is the treatment? How can treat this pathology disease in general? Currently, there are not cure for neurodegeneration. For each of the neurodegeneration disease, there are specific drugs that can be used to minimize their symptom. For example, donepezil for and memantine for Alzheimer's disease, and L-DOPA for Parkinson, and result for ALS. That we can confirm that it's not cure for neurodegenerative or degenerative disease. Okay, why the brain is vulnerable to injury? This is some basic information about this. For example, neurons are post-nicotic cells, little anaerobic capacity, dependence on glucose, high metabolite rate, and many substances go directly to the brain via inhalation. Okay, this is the main effect of peripheral gain contribute to brain injury. If you see that this picture present that the MET-induced organ malfunction induced brain pathology. For example, we can see hypoxia, ischemia, and systemic inflammation, and the gas of ammonia. Sorry, but we have different systemic inflammation that we can contribute to produce the problem brain injury in general, as lungs, heart, liver, muscle, kidney, etc. Okay, today I present the problem of Alzheimer's disease because the World Health Organization, WHO, has estimated that 35 million people are currently living with dementia worldwide, which will be further increase in 2030 to 75 million, and into 2050 at 135 million people. I think that it's very, very serious problem in the world that we can remark that it was that it is 600 neurological disease. For your information, the Center of Disease Control and Prevention said that the number with Alzheimer's disease, the dementia, in 2021, the total is 6 million people. This is the effect of COVID-19 pandemic on death from Alzheimer's disease. Preliminary report from the CDC indicate that there were at least 42,000 more deaths from Alzheimer's disease and other dementia in 2020, compared with the average of the 5 years before. I think that it's very, very important, and it's very, very seriously disease in the world. Okay, now I present some information, the basic information of Alzheimer's disease. If you see now, that is the comparative between the healthy brain and Alzheimer's disease. In the case of Alzheimer's disease, if you see that we have this contraction of neuronal cells and the problem to produce amyloid plaque and the protein of tau. Okay, this is the balance between healthy brain and Alzheimer's disease. If you know that this balance in the case of metal overload, for example, aluminium, and in the case I publish more paper in this subject, problem of amyloid beta plaque formation, inflammatory response, and the protein tau. I think that it's very, very difference between healthy brain and Alzheimer's disease. Okay, I start my hypothesis in my research in my laboratory from this picture. If you see that the production of free radicals from different factors, for example, metals or inflammation or problem of mitochondria, this introduce the synaptic dysfunction, and I think that is the key of problem of Alzheimer's disease. Okay, this picture presents the problem of the different factor of Alzheimer's disease in detail. For example, if you see that the problem of structure of neuron cells as astrocytes and microglia, we have dysfunction in the neurotransmission between postsynaptic and presynaptic membrane between two neurons. For example, we have problem of aluminium effect, etc. Okay, what is the different factor? We can see the vascular risk factor, physical, genetic, the level of education, family history, the age, and the gender, and in particularly environmental factor as aluminium or aluminium toxicity. Okay, what about the treatment? About the treatment, we have synthetic treatment, and we have synthetic treatment drug. We have synthetic no drug therapy, and we have phytotherapeutic treatment. Okay, now if you see about the drugs, we have acetylcholinesterase inhibitor, anti-inflammatory. We have other antioxidant as vitamin B12 and other of, for example, folic acid. This is the different drug or synthetic drug to treat the problem of Alzheimer's disease. About synthetic drug treatment, we can improvement of cognitive function, mood, behavioral disorders in the old people, to reducing stress, helping physically and mentally, preservation of links and exchange socio, and the preservation of functional anatomy. This is in general. About the synthetic drug treatment. Okay. In conclusion, we can say that the pharmacological treatment, none of the pharmacological treatment, I mean drug, available today for Alzheimer's dementia, slow or stop the damage and destruction of neuron that cause Alzheimer's symptom and make the disease fatal. The Food and Drug Administration from US has approved that drugs for treatment of Alzheimer. We can present some drug. For example, we have donepezil, approved in 1996. We have rivastigmine, galantamine. This is the cholinesterase inhibitor. Okay, but about the the type of drug works, we have memantine and we have donepezil. Okay, we confirm that no drug are specifically approved by the Food Administration drug to treat this pathology. For example, what is the different phytotherapy in the world? If you see, we can see we have Ayurveda medicine, we have Chinese traditional medicine, we have European herb medicine, we have African and South American traditional medicine. These different medicine can stop or inhibit the problem of Alzheimer's disease, and we have prophetic medicine. Okay, the plant and natural source from the basic of today modern medicine. Okay, I can remark that the plant world is full for precious and virtual, from which and man draw not only his food but also active substance. That no drug that modify or prevent the propriety of Alzheimer's disease. Therefore, there is clear that we need for new and more effective development to treat or modify the progression of Alzheimer's disease. This is different biological activities or different treatment of different medicinal plant as hypotoxicity, or renal damage, cytotoxicity, cancer development, and etc. About medicinal plant which were rich of polyphenol. If you see in this picture, we can decrease the brain level of amyloid peptide, and we can reduce oxidative stress. This is very important to treatment of Alzheimer's disease. This is a different medicinal plant to treat this problem, Alzheimer's disease, as ambiliboate, as ginkgo biloba, as dorstina. This is the different medicinal plant that which used in the treatment of this disease. I mean Alzheimer disease. Okay, my hypothesis is from this picture. We can block, or we can inhibit, or we can stop the free radicals or the production of or free radicals. Automatically we can stop the synaptic dysfunction. I mean, we can inhibit problem of Alzheimer disease. In my country, Algeria is in the north. It's in the north of Africa. We have 4 seasons. We have a very big desert, I mean Sahara, and we are very rich in medicinal plant. We are very close to Europe, and we are Mediterranean country. For experimental animals, I present some data of my PhD student. For example, in my laboratory, we try to treat this problem, this Alzheimer disease, by different plants. If you see, we use the curcumin, we use the passiflora, we use the pomegranate, we use daughter plant, and we find the positive data about memory and about Alzheimer disease. Okay, I can now present the data of my recent PhD student, that we used the mix of 2 plants. It's about mushroom, if you know the mushroom, and the curcumin. This is under international project between my laboratory of Algeria and laboratory from Vietnam, from Hanoi, Vietnam Academy of Science and Technology. This is Institute of Natural Product, that we use the mix of two substances to treat this problem in vivo. Okay, this is the complex. It's about 1,3-beta-glucan curcumin. This is the mix of mushroom and curcumin, and we try to treat this problem. I inform you that I remind you that we recently published the paper in this subject. Okay, the first parameters, it's very simple. It's about phytochemical about this 2 plant, medicinal mushroom and curcumin, and we analyze this by FTIR, and I analyze it by HPLC. Okay, this is the different neurological test about Alzheimer disease. If you see, we have locomotor activity, problem of anxiety, and we have swimming, forced swimming, and we have the maze, and we have Hayden platform about about Alzheimer disease. Okay, other tests in my laboratory that we use. This is the maze, the maze called plus double H in double H, and we can calculate the memory of mouses. That we use groups, Alzheimer groups, and control groups, and Alzheimer treated by 3 different doses about mushroom and curcumin. The third parameters, it's after sacrifice. We can analyze some biochemical analysis and histological study. Now this is some data of our research in my laboratory. If you see that about locomotor activity in Alzheimer model, we have 4 phases or 4 steps. Each steps it's for 5 minutes, and we can calculate it by score the locomotor activity in different group: Alzheimer and control, and Alzheimer treated by 2 or 3 doses. In the 5th days, this is the test locomotor activity as the curiosity or test of curiosity. It's the same. And we have anxiety, and we have the test of Elevate Plus. In conclusion, I can see, for example, this is the test of Petzold, forced swimming, and we can in the test, because after 4 days of learning period, we find that our product, it's very important of memory. This is the picture of the 5th day about spatial memory of work. And we have for test of Maurice water maze. Maurice water maze, it's after 4 days of learning, we can calculate it, what is the difference. If you see now that the treatment by 2 doses, the first and the second, it's very approach of control comparative to Alzheimer groups. Same of Maurice Mays Alzheimer. This is MSR, and we can conclude that our product is very important to ameliorate problem of memory. About histological parameters, we studied the cerebral cortex and kidney and liver. For example, about the brain, this is difference between control and Alzheimer disease. We have a very big difference between this picture, Alzheimer and control. This is the treatment. Treatment by the first doses is 200 milligram by kg, and the second is 250 milligram by kg in the second treatment. This is about kidney. Sorry, it's about liver. We have a different picture about treatment at Parkinson, and same. This is the kidney, okay? And if you have question, I can answer you after my presentation. In the conclusion, I can say that the current treatment do not be beyond the inhibition of acetylcholinesterase, while herb medicine present by some medicine plant of the problem of oxidative stress and thus reduce tissue damage. We can in the future to confirm our data that we need, or we have to do some technical in vitro, because the cause neurodegenerative disease in Alzheimer disease and Parkinson disease by phytotherapy using other Algerian medicinal plant. This is my laboratory. It's in English and in French and in Arabic. And sorry if my pronunciation, because we speak Arabic and we speak French. This is the picture of my team in my laboratory, and this is picture of my city. It's Mostaganem. Thank you for your attention.
Thank you, Doctor, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and presentation. Now let's move on to the next presentation. The next presenter is Dr. Amir Choram from Fax University, Tunisia. Hello, Dr. Amir. I'm ready with your presentation.
Hello everybody. Okay, I will see now.
Okay, Doctor, now I'm making as a presenter. Kindly click on the screen button to share your screen.
Yes, thanks.
I made you as a presenter. Yeah, your screen is visible now. You can please make it in a presentation mode and start your presentation.
Okay, is it clear now?
Yeah, it is perfect now. You can start your presentation.
Okay, thanks. Okay, thank you for this opportunity. My name is Amir Sherm from Tunisia. My communication is entitled Effects of Caffeine Ingestion on Psychomotor State and Oxidative Stress Markers After an 8-kilometer Run Competition in Sleep-Deprived Recreational Runners. The plan of this communication, we have 4 sections. The first section is the introduction and problematics. Then we have material and methods. After that, you have results and discussion. And finally, we have the conclusions. About the introduction, we will clear the principal parameters of this study. So we begin with caffeine. What is caffeine? Caffeine is the most consumed psychoactive substance in the world. We could find caffeine in coffee and tea, also in chocolate and in soda. Also, caffeine is a powerful central nervous system stimulant. And caffeine could lead to improved alertness, strength, and endurance. In addition, it could lead to improved sports performance. The second parameter is sleep. We have 4 kinds of sleep. We have very light sleep, deep sleep, and paradoxal sleep. So sleep is a process of repair and homeostatic regulation. Also, the adequate sleep is between 6 and 8 hours per night, and it is fundamental for health. However, sleep deprivation could alter physical, mental, and cognitive state. And these effects are more pronounced when we have total sleep deprivation, TSD, that is exceeding 24 hours of sleep deprivation. The third parameter is oxidative stress. So what is oxidative stress? It is the main reason for the deterioration of cells. It is also an imbalance between the production of free radicals, FR, and the antioxidant capacities of the body. The defense system of our body have enzymatic antioxidants such as SOD and GPX, etc., and non-enzymatic antioxidants such as GSH and uric acid, etc. And this defense system could lead to the trapping of inhibition or the inhibition of free radicals production, example in aerobic exercise. The problematics of the study. Although caffeine is a commonly used aid in athletic performance, especially among top athletes, few arguments exist to justify its restorative effect on psychocognitive parameters and those of oxidative stress after total sleep deprivation. We have 2 questions. The first question, could caffeine supplementation prevent the deleterious effect of total sleep deprivation following aerobic exercise? The second question, does caffeine exert an antioxidant effect during this type of exercise? The first section is material, and the second section is material and methods. And we have four 4 sections. We begin with the objective of the study, then the participants, then we will see the protocols, and finally the tests. For the aim of this study, we have to analyze the effect of the ingestion of 5 milligrams of caffeine before the race of 8 kilometers, and this is after reference night or total sleep deprivation night. And we will analyze this effect in 4 parameters: the physical parameter, and secondly the mood and the cognition, and finally the oxidative stress parameter. For the participants of the study, we have 10 participants. Their age is about 21 years. Their mass is about 66 kilograms. The height is around 1 meter 76, and the body mass index is about 21, and the VO2 max is about 51. About the protocol of the study, here we have 4 sessions. We have 2 sessions after a reference night and 2 sessions after a total sleep deprivation night. Reference night RN and total sleep deprivation night TSD. And the reference night, participants will sleep from 22:30 to 7 hours, and for the total sleep deprivation night, participants will not sleep. After that, at 7:30, participants will have capsules containing 5 milligrams of caffeine, and in 8:30 we will realize the blood intake and the psychomotor tests. At 9 hours, participants will begin the test of 8-kilometer run, and at the end of exercise we have performances, and we will realize again the blood intake and the psychomotor tests. The tests used in this study, we have firstly the 8-kilometer running around the 400-meter outdoor athletic track of the university. The second tests after the run, we have 2 tests. We have the feeling scale tests, FS, and the Hooper test. For the feeling scale tests, participants will send a paper for giving the feeling of after the running. And for the Hooper tests, they give a subject evaluation of the study of the run, about sleeping, about the stress, about the fatigue, and about the pain. The third tests used in the study, we have the digit constellation test in the left and the reaction time test in the right. For the digit constellation test, participants will constellate every numbers of 3 digits in 1 minute. And for the reaction time tests, participants will push the space of the microcomputer of the computer as quickly as possible for 10 times. Finally, the test of the final test is the collection and the treatment of blood samples. Here we collect all the blood samples, and we will take them in the laboratory of medicine to analyze them and give the results. The third section now, the results and discussion. For the results, we will see, as we said, the physical parameters, the mood, the cognition, and finally the oxidative stress parameters. For the physical parameter, the performance of 8-kilometer run following the ingestion of caffeine. We have here in blue the placebo group, and in red, the caffeine group. Here in the left, we have after reference night, and in the right, after the total sleep deprivation night. As we see here, the performance of the caffeine group is best than the placebo group, following reference night and also following total sleep deprivation night. So the administration of 5 mg caffeine significantly improved the running performance after a reference night by 2.8%, as well as after total sleep deprivation night by 4.9% compared to the placebo group. Also, we can see in this figure that there is no significant difference between the performance of the control group following reference night and that of the supplemented group after total sleep deprivation night. So, caffeine seems to restore the physical performance which is affected by total sleep deprivation. This recovery could be due to the antagonistic effect of caffeine on adenosine receptors, which could improve wakefulness and reduce drowsiness. We pass now to the psychological state. We have two tests. The first test is the test of feeling scale. As we see here, placebo group in blue and the caffeine group in red. We can see the caffeine group after the reference night and after the total sleep deprivation night is usually best about the feeling scale is usually best than the placebo group. So we could conclude that the consumption of 5 mg caffeine increased feelings of well-being after running competition following reference night and total sleep deprivation night. Also, caffeine could be effective in minimizing negative psychological effects induced by sleep loss conditions. The second psychological test is the test of Hopper. We have 4 parameters. We have stress, pain, fatigue, and sleep. About the stress, as we see in this table, the stress after exercise is minimized from 4 to 3.5. Also, the fatigue is reduced from 6.3 to 5.4. So, caffeine consumed after total sleep deprivation reduced the feeling of stress by 18.3% and the feeling of fatigue by 14.3%. We can say that the analgesic properties of caffeine seem to help delay fatigue after reference night or TSD, which improves psychomotor state and physical performance. Now we pass for the cognitive state. We have two tests, the test of reaction time and the test of digit constellation of numbers. For the test of reaction time, we have here after reference night and after total sleep deprivation night, in blue placebo and in red the caffeine group. As we see here, the caffeine group have best times before in the reference night and after total sleep deprivation night. For the digit constellations, the numbers concealed for the caffeine group are greater than the placebo group following the reference night and also following the total sleep deprivation night in the right. So we could conclude that caffeine consumption led to faster reaction and better attention after reference night and also after total sleep deprivation night. This beneficial effect of caffeine on the psychic state could positively affect the cognitive states of athletes despite their alteration following total sleep deprivation. And here we have the fourth parameter, which is oxidative stress state. We have 4 parameters, GPX, GSH, and SOD, and also the peroxidation parameter of MDR. As we see here on the table, these parameters are not altered from caffeine, but they are altered from the exercise and from the condition of sleep. For example, GPX is reduced from 8.4 to 6.7 following the condition of sleeping. Also, the MDR is elevated from 5.2 to 6.1 following the exercise, and we have not the effect of caffeine. So we could see that caffeine does not appear to improve oxidative stress status after reference night or total sleep deprivation. And the observed changes were influenced only by exercise and sleep conditions. In this way, current experimental evidence in animals suggests that sleep deprivation promotes oxidative stress by lowering antioxidant defenses. And the conclusion, from this study, we could conclude that 5 milligrams of caffeine before the running of 8 kilometers have a good effect on performance. It improves significantly the performance. Also, it improves the mood state and improves the condition of athletes. But for the oxidative stress, there is no effect of caffeine on the oxidative stress. This study, we have two articles, two papers that was published in the biological rhythm research. And finally, thank you for your attention.
Thank you, Dr. Amir. Does anyone having any queries? I hope there are no queries. Now, once again, thank you, Dr. Amir, for your presence and presentation.
Thank you
Now
very much for this opportunity. Thank you.
Oh, thank you. Now let's move on to the next presentation. The next presenter is Dr. Tanjiala from Southeast University, China. Hello, Dr. Tanjiala. Are you ready with your presentation?
Yes. Hello, everyone. Yes, my presentation is ready.
Okay, now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Okay.
Yeah, your screen is visible now. Make it in a presentation mode and start your presentation.
Okay. Hello, everyone. I'm Dr. Tanzila from Southeast University, China. Today, I am going to present about pH responsive gold BSA encapsulated exosome for cancer diagnosis and targeted drug delivery. Sorry. Cancer is leading cause of death, and there are many drug delivery carriers like cationic polymers and many other. But there are some limitations for traditional drug carriers, like they release rapid and burst drug release, require more quantity of drug, rapid immune clearance, poor availability to tumor site, off-site toxicity, cannot cross blood-brain barriers. Exosomes are attention-grabbing therapeutic tools for cancer therapy, and they are very common nowadays. Exosomes have some specialties which make it very common for cancer drug delivery, as they are nano-sized, biocompatible, can cross blood-brain barriers, carry information exclusive to their ancestor cells, non-immunogenic lipid bilayer, stabilize circulation, promote efficient targeting and cellular uptake. As here we can see in this picture, exosome can pass from blood circulation easily and target the tumor cells. Moreover, exosome can be used as diagnosis for cancer, like we can take exosome from blood and diagnose by biopsy. Next is material method of our experiment. First of all, we have synthesized gold BSA nanoclusters from gold salt and isolate them from cells by freeze and thaw method. Then we characterize these gold BSA nanoclusters. After characterization, we confirm that cancer cells can biosynthesize gold BSA nanocluster. Then we have synthesized in situ biosynthesized exosomes in cancer cells by passive loading and isolate from cancer cells by ultracentrifugation and characterize their size, their surface charge, their doxorubicin contents, etc. After that, we use these isolated exosomes for to check as bioimaging for toxicity against cancer cells in vitro. Here are our results. First of all, we perform MTT assay to check the toxicity of our substance like BSA, gold and gold BSA. We used BSA, we observed it is stable. It is not toxic, not for cancer cells, even not for normal cells. Gold is little bit toxic for cancer cells, but it is not toxic for normal cells. But gold BSA is very toxic for cancer cells like UAT7 brain cancer cells, but it is biocompatible for allo2 cells, normal cells, healthy cells. Next, we used fluorescence imaging of these free substances to confirm that cancer cell can uptake gold BSA or not. We observed cancer cells can easily uptake synthesize gold BSA, which confirm it. Cancer cells are producing gold BSA nanoclusters. After confirming, we have isolated gold BSA nanoclusters by freeze and thaw method. We treated the cells for 24 hours with gold BSA. After that, we removed cells from cell culture dish and centrifuged it and freeze and thaw 5 times. Then centrifuge for 10 minutes at 800 xg to isolate gold BSA nanoclusters. After that, we have characterized them. In this image, these are TEM images of gold BSA nanoclusters at different scales. This is the average size of gold nanoclusters, gold BSA nanoclusters. We have observed 3.3 nanometer average size of gold BSA nanoclusters. Next, this is the elemental characterization of gold BSA nanoclusters. Here we can see the peaks with gold, which confirm the presence of gold in sample. Here is the absorbance of gold BSA. The peak value of absorbance was at 290 nanometer. And next is fluorescence intensity, excitation, emission. The excitation was at 490 nanometer and emission wavelength was at 580 nanometer. Next, after confirmation that brain cancer cells can biosynthesize nanoclusters, we isolate exosome from treated cells. We treated the U87 cells with gold BSA and doxorubicin. After 48 hours, we collect cell culture medium of that treated cells separately and combine. We also isolated blank exosomes. Here is procedure for exosome isolation. After collection cell culture medium, we have centrifuged it at 800 xg for 10 minutes to remove dead cells. After that, at 2000 for 20 minutes to remove cell debris. And at next step, 10,000 for 30 minutes to remove free proteins. Next, we filter these supernatant to remove free RNA or DNA. Next, we ultracentrifugate this supernatant at for 90 minutes and remove the supernatant, and we save pellet and dissolve the pellet in PBS and save it for future characterization and treatments. Here is characterization of exosomes derived from treated and non-treated U87 cells. First one is team image of exosomes. First, this one is blank exosome, like derived from non-treated U87 cells. Second one is derived from U87 cells treated with BSA. This one treated with gold BSA docs. In team image, we can observe exosome derived from treated U87 cells have black dense dots, which confirm its loading. However, the blank or BSA loaded exosome have no dots. In next tree, we have gold nanocluster, which show dark dense particles in exosomes. In next, these are the confocal images of exosomes. This one is fluorescence and this is overlay. We can see the loaded exosome show fluorescence, a strong fluorescence, but the control and BSA treated exosome don't have any fluorescence. So it is confirmed that exosomes are loaded with gold nanoclusters and doxorubicin. Here is the total protein content of exosomes. We have by here the total protein concentration of different groups. We observed the treated cells release more exosomes and non-treated cells release less exosomes. As when we treated the cells with doxorubicin and gold BSA, it was under stress. Under stress, cells release more quantity of exosomes. Here is average size of exosomes. First one is size of blank exosome, and second one is loaded exosomes. There is only minor difference of size. There is no major difference between loaded and unloaded exosome because exosomes have empty space. Then and the loaded material is inside the empty space. It didn't change the size of exosome. Here is doxorubicin concentration in exosome. We measured it by HPLC. and the average doxorubicin content was 7.6 nanogram per microgram of exosome. Here is elemental characterization of exosomes. Here is two peaks of gold which confirm the presence of gold inside exosomes. Here is pH responsive drug release from exosome loaded with gold BSA nanoclusters and doxorubicin. We use this method to calculate the pH responsive drug release. We use dialysis bag. We put our material in dialysis bag and put it releasing medium like PBS with different pH levels. We collect sample after different time intervals and measure its doxorubicin concentration. We observed exosome release their doxorubicin at acidic pH, which is beneficial for treatment of cancer as the pH at cancer size is acidic. However, in normal cells or healthy tissues, pH is 7 something. And we observed that exosome did not release quickly. It release sustained release. Like we can observe after 72 hours, the concentration of doxorubicin is continually increasing. It not remains same. Its mean exosome release drug sustainable. Like it can circulate or release drug for long time, remain in body for long time. Here is bioimaging of different groups of exosomes. First one is control. We can see there is no fluorescence. Second one is exosome with gold. It has less fluorescence than gold BSA and gold BSA docs. Gold BSA is more fluorescence because BSA make it stabilized and more gold nanocluster formation occur. And doxorubicin have very strong fluorescence in cancer cells, but in LO2 it has no fluorescence. Thus, we can conclude that gold BSA nanoclusters and doxorubicin loaded exosomes are compatible for normal cells. So here is, can you see MTT this side? Okay. Here is MTT assay to calculate the cell viability against different groups of exosomes and other free substances. We have used similar concentration of doxorubicin even it was free with gold BSA nanoclusters or even with exosomes. We use same concentration of doxorubicin. We observe that similar concentration of doxorubicin is less toxic. Free doxorubicin is less toxic than doxorubicin encapsulated in exosomes along with gold BSA nanoclusters. But it is not toxic for normal cells. Next is we use photodynamic therapy. We used we do phototherapy of different groups of exosome at 0.6 concentration nanogram concentration of doxorubicin. We observed when we used phototherapy, it killed more cells than alone exosomes. So exosomes can also facilitate phototherapy. Here is ROS accumulation in cancer cells. Here are U87 cells uh, treated with DCFDH assay to calculate the ROS accumulation in cancer cells. We observe gold BSA doxorubicin loaded exosome have more ROS level than only gold BSA loaded exosomes. And both have, when we applied phototherapy, it has more accumulation of ROS than without phototherapy. Here is nucleus staining by DAPI to observe cell apoptosis. First group is control exosome. We can observe in control and exosomes nucleus material is organized. But when we treated it with exosome loaded with gold BSA nanoclusters, DNA start condense. After that, with doxorubicin, it start floating. And when we use phototherapy, it start floating and nucleus membrane removed and DNA material spread. Here is cancer cell apoptosis by flow cytometry. We treated cancer cells with gold BSA and gold BSA doxorubicin with or without phototherapy. We observe exosome loaded with gold BSA and doxorubicin are more toxic for cancer cells. Conclusion. Cancer exocytosed exosomes were loaded with bio-assembled gold BSA nanoclusters. These nanoclusters were bioresponsive self-assembly of small carriers for targeted chemotherapy. Engineered exosome showed strong fluorescence in cancer cells but not in normal cells. This concludes that we can use these exosomes for cancer diagnosis, early cancer diagnosis by bioimaging, as they only give fluorescence in cancer tissue, but not in normal tissues. Finally, it is engineered exosomes showed cytotoxicity against cancer cells but safe to normal cells. Thus, we can use these exosomes for cancer therapy efficiently. Here is most beautiful view of my university, Southeast University. Thank you for your attention. Any comment, question, and suggestions are highly acknowledged.
Thank you, Dr. Chanila, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and
You are
presentation.
welcome. You are
Now let's
welcome.
move
Thank
on
you
to the
for your
next presentation. Thank you, Dr. Tanjila. Now let's
you
move
are
on to the next
welcome.
presentation. The next presenter is Dr. Dufera Rikichu Terifa from Wolga University, Ethiopia. Hello, Dr. Dufera. Are you ready with your presentation? Hello, Dr. Dufera.
Ma'am, can I present my paper?
Hello, Dr. Dufera. Hello, Dr. Dufera. Okay, we'll contact Dufera in a while, and now we'll go ahead with the session. The next Chetan Kumar Dubey from Career Point University, Kota, India. Hello, Dr. Chetan Dubey. Are you ready with the presentation?
Yes, ma'am, yes.
Okay, now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Can I start, ma'am?
Okay, okay, Dr. Chetan, your screen is visible. Can you make it is in a full screen mode and start your presentation?
Okay. Firstly, I'll thankful to our organized committee give me a chance to present my paper here. Am I audible? Am I audible, ma'am? Am I audible, ma'am? The topic of my presentation is screening of anti-anxiety activity of gingerol on nodates. Gingerol is a main active constituent present in ginger. It is very helpful and very key medical role in therapy of very psychoactive substances, psychoactive disorders. Now, what is anxiety? First, we discuss about anxiety. Anxiety is a disorder related to mental health, diagnosed with leads to excessive nervousness, fever, apprehension, and worry. Anxiety is a normal and often healthy emotions. However, when peers regularly real disappointment level of anxiety, it might become medical a disorder. Firstly, it is not a disorder. When it is long-term anxiety, we have it is feel like a disorder. These are the major symptoms of anxiety, like excessive anxiety and worry, increase the muscles ache and soreness, impair the concentration, irritability, fatigue, restlessness, difficulty in sleeping. These are the major symptoms. By the help of we can recognize the patient. This is an anxiety patient. Next, about epidemiology, worldwide 3.6% of population worldwide, near about 264 million individuals worldwide have anxiety. Additionally, more the females are more prone than male having anxiety. The females are more affected than males through anxiety. These are some etiological factors like genetic factor, environmental factor, like some childhood trauma. These are responsible for the development of the anxiety. The unrecognized medical conditions like some illness, uh, long time illness also responsible for anxiety. Some medical substances, some OTC medicines, some herbal medicaments, some abused substances like hemp, charas, ganja, bhang. These are also produce the anxiety. There are some when we are come in part of pharmacology. There are some neurotransmitters like GABA, gamma aminobutyric acid, serotonin, norepinephrine are the major neurotransmitters that play a key role in the development of the uh, anxiety. The low level of GABA aminobutyric acid at presynaptic neuron junctions are responsible for the development of the anxiety. And some norepinephrine, these are located into locus coeruleus part of the brain. Norepinephrine also have have major neurotransmitter play a role of development of anxiety. These some anxiogenic agents are having or decrease the GABA level at GABA receptor and leads to the nerve excitability and produce the anxiety. some medicines are used in treatment of the anxiety, like benzodiazepines, diazepam, oxazepam, lorazepam. These are the drugs which are used in uh, treatment of the anxiety. Some azopirons like buspirons, some sedative, sorry, some sedative hypnotics like hydroxyzylene, propranolol. Sorry, some other medicines like tricyclic antidepressant, chlorphenamide. These are the important drugs used in treatment of anti-anxiety. My objective of study, various pharmaceutical agents are used in therapy of anxiety, but having some uh, side effect like sexual dysfunction, restlessness, nervousness, insomnia, fatigue, sedative, and dizziness. So I'm planning to initiate some medicinal preparations that can be used in the treatment of anxiety. So that's why I choose some herbal compounds for systering of anti-anxiety activity. For this purpose, first we select the we design one experiment. First we started with the toxicity studies like by the help of OECD guideline 423. In this study, we divide animals in two groups. One group having 3 HG rates and second having 3 HG rates. First one is control group. We gave 1 ml per kg body weight, the standard controls 1% CMC solution. And second group, we gave the extract of gingiber officinal, and third group is we give the hydroalcoholic extract of hibiscus rosea sinensis. Is the major key role. And this protocol also approved by the institutional animal ethical committee, and the animals were fed by the following compound and assess the following parameters. Like these are the results of our studies, like parameters like appearance, activity, fur coat, mucous membrane, body orifices, eye, opacities, urinations, unusual respirations, pupil size, excessive grooming, bile excretion. We studied the whole 14 days of these parameters and found the normals in both are the plant. And we said that about the data, that no mortality were seen in 7 days or 14 days. These are the parameters like body weight we observe in 0 days, 7 days, and 14 days. This is the control group of animals having the similar growth in body weight, and there is normal parameters. Their mean value of range 187 gram to 206 gram. Now, another point, we also record the food consumptions of animal and water consumptions. These are the three graphs. So blue represent the control group, and the purple color is for the HRS extract, and green is represent the gingiber officinalis extract. This is the initial first day body weight food consumption report. This is the at 7 days food consumption report, and is final day after 14 days. So initially we say that the food consumption of animals are increased when we increase the food. Some water consumption parameters like that. The water consumption also decrease in medium in after 7 days, but after 14 days it is normal. So there is no evidence of any toxicities in food and water consumption. In this toxicity study, we also assess the same hematological analysis like hemoglobin content, RBC count, MC mean peripheral volume count, MCSC, MCA, TCV, TLC, granulocyte, lymphocytes, and monocytes. When we are comparing with the control, the data we achieved very normal. In HRS group, some lymphocyte count also increased due to the some antigen antibody reactions. Now these are organ weight data, like we autopsize the animals and weigh the liver weight, lungs weight, kidney weight, left kidney weight, right kidney weight, brain weight, and we are comparing with control, somewhat decrease the weight of liver comparatively, and lungs weight is about to normal in both groups. The weight is somewhat increased because it's very high dose, 2000 gram per kg weight. And after we also done the histopathological studies, is showing the normal value of size, the cellular, the cell wall and other micronucleus of the cell are normal in both is the control brain and is the alcoholic acid brain, heart and heart. So both are the having normals in. There is no significant change observed in this study. About the liver and lungs, ovary, they as normal are seen. Is the we assess the experimental design for anti-anxiety activity. We choose the we choose the two dose for that after the acute toxicity study, 200 mg per kg weight body weight and 400 mg body per weight. We divided into uh, three groups: control, standard, four group: control, standard, and hydroxyl rhizomes and hydroxyl of by 400 mg per kg weight. This also approved by the CPC IEC PhD 2014. Now we assess the model for eliminated plus minus model by reference Vogel et al. Book. This figure is showing the number of entries in open arm. This is the data of control. This is standard. When we are compare with the standard like diazepam, the 400 mg per kg dose of rhizomes with similar is somewhat less effective, but when you comparing with control, it having showing the anti-anxiety effect. And also the elevated plus minus time, the time spent in closed arm also decrease with comparison to when we are compared with the control. These, if you observe the hydroalkyl state of rhizome during of extract at dose of 400 mg effective as a standard drug in the anti-anxiety activity in mice by increasing time spent open arm entries and into the EPM models. Like when second model we are take like open field test, is the effect of diazepam and hydroalkyl rhizomes. These the four groups like control, standard diazepam 2 mg per kg, 200 mg per kg test 2, 400 mg per kg. This is the ambulation time, number of square are crossed. When we compare the control, the ambulation capacity of animal increase when we are increasing the dose. The ambulation period also increase, and as Edo said, is the rearing activity also increase and activity in center are also increase. Fecal discharge is decreased. These are the differences. Thank you. If any questions, is there any questions?
Thank you, Dr. Chetan, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and presentation. Now let's move on
Thank
to the next presentation. Hello, Dr. Defera. Are you ready with your presentation? Hello, Dr. Defera. Okay, Dr. DeFera, now I'm making you as a presenter. Kindly click on the screen button to share your screen. Hello, Dr. DeFera. I hope Dr. DeFera has some technical issues. Now let's move on to the next presenter. The next presenter is Dr. Moifo Kute Thomas Barland from University of Ude, Cameroon. Hello, Dr. Thomas. Are you ready with your presentation?
Yeah.
Okay, Doctor. Now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Okay. I think it's okay.
Yeah, your screen is visible now. Can you make it in your presentation mode and start your presentation?
Okay.
In a slideshow, you can select the presentation mode on the top.
Okay. So, let me. Okay. Can I start?
Yeah, you can start.
Okay. Thank you.
Just we are requesting you to make it in a presentation mode. That's it. That
Okay.
is full screen mode.
Okay. Thank you for the opportunity you are giving me to present the part of my research work, PhD research work. I'm not yet doctor, so I'm still a PhD student. Yeah. So the work I'm going to present to you is entitled New Antibacterial Phenolic Compound Isolated from Ashlanthus bruneus plant, which is an hemiparasitic parasite plant. So the outline is as follows. We start by a general introduction, following by the literature review. After the result and discussion, I will finish by conclusion and perspective. So microorganisms are widely distributed in the environment and the majority in a big terrestrial and aquatic environment. Indeed, water, apart from its importance in the daily life of men for nutritional and practical needs, such as body hygiene, household tasks, is also a medium for waterborne human pathogen dissemination. So access to drinking water is an obstacle to improving the health of population despite a water supply and sanitation program that have been put in place since 2.4 billion people still live without adequate sanitation facilities, with sub-Saharan Africa being the most greatest, according to WHO 2019 and 2015. So, contaminated drinking water, uh, when used, is in the preparation of food, can be the source of foodborne disease through consumption of microorganisms. So you are seeing this woman with very dirty place which is cooking, and you are seeing this picture, the left picture where the bacterial strain are distributed in water. So some water and foodborne diseases are cholera, infectious hepatitis, giardiasis, amoebiasis, diarrhea, and typhoid. So typhoid is an acute illness associated with fever caused by Salmonella enterica serotype bacteria. According to WHO, 22 billion documented cases with 200,000 fatalities are reported annually worldwide. Further estimates suggests that 88% of that burden is attributable to unsafe water supply, sanitation, and hygiene, and is mostly concentrated on children in developing countries. So, these are some medicine currently used to cure typhoid. You can see chloramphenicol, trimethoprim, sulfamethoxazole, ampicillin, gatifloxacin, and ciprofloxacin. So, the treatment already exists, but what is the problem? The problem is that in recent years, many research have shown a strong emergence of strains of of microorganisms that are multi-resistant to this antibiotic, making them becoming increasingly ineffective. So this difficulty is compounded by other factors such as the availability of this molecule in terms of cost and stock, especially for developing countries, as well as their misuse, which complicate the treatment. So the challenge is to find a new active molecule that are more accessible to local population and capable to overcoming this problem of bacterial resistance. This is why knowing the demonstrated potential of medicinal plant at the traditional level in the treatment of disease and also in the discovery of new lead molecules, future drugs, we chose and investigated Agelanthus breneus with the hypothesis that it could contain antibacterial molecule. So the choice of this plant was motivated by the fact that some species of Agelanthus genus are frequently used in Cameroonian traditional medicine to treat several diseases such as diabetes, abdominal pain, urinary tract infections, malaria, cancer, parasitic and bacterial diseases. Also, crude extract from some species have exhibited vasorelaxant, anti-inflammatory, antiplasmodial, and antibacterial activities, according to Boli, Traore, and Bulda, respectively, in 2019, 2004, and 2012. So, the main objective of this study was to isolate and perform the antibacterial activities of chemical component from Agelanthus breneus. More precisely, extract, isolate, and purify and characterize the secondary metabolite from the above mentioned plant. Also, they evaluate the antibacterial activities as well as those of extract from which they are derived. So literature review on the plant species investigated. So you can see in the right side the pictures of the plant. You can see the flowers, the leaves, and the stems. So Agelanthus breneus is a hemiparasitic plant belonging to the Laurentaceae family, which comprise more than 70 genera and about 900 species growing in Africa, Asia, America, and Europe. In Cameroon, the Rhotaceae family is represented by 26 species distributed in 7 genera. So the 7 genera are Agelanthus, the species, the genera we investigated, Englirina, Globimetula, and others. So some species of the Agelanthus genus are frequently used in Cameroonian traditional medicine to treat several diseases such as diabetes, abdominal pain, and bacterial diseases. So the table here, in this table, some authors have tested the crude extract of some species of the family Laurentaceae, and the results are present. So this extract, the first extract, has excellent anti-inflammatory properties, and the second antibacterial activity against Escherichia coli, Staphylococcus aureus, and the last one. So the antibacterial potential of some
Hello, Dr.
species.
Thomas.
Yeah,
Hello, Dr. Thomas.
yeah,
Your PPT is not changing. You are in the first slide still.
it's okay.
No, not yet.
SA5.
Which one, please?
May I know the which slide you are explaining?
This one?
Your one?
Okay.
In
So,
which? Hello, Dr. Thomas. May I know which
yeah,
slide you are explaining?
this one. I was explaining this one. This slide.
May I know the slide number?
Oh, the slide were not numbered. I don't. Sorry about that.
Because we are seeing the first slide still, the first introduction slide.
Okay. So it is slide number 12
Yeah, it is
here.
perfect now. Okay, it is perfect. You can start.
Okay. So I was trying to say that the antibacterial potential of some species of Laurentiaceae have been demonstrated in the literature. So that was one of our best motivation in our study. From some species, chemical compounds were isolated. As you can see, we have just illustrated three of those compounds. We have two pyrrole and one flavonoid. And the first pyrrole has excellent anti-plasmodial activities with IC50 of 2.4 and 4.9 millimeters. And the second exert relaxant effect on preconstituted rat. And the last one exert moderate activity against the bacteria Pseudomonas aeruginosa with MIC of an MBC value of 250 microgram per milliliters. So let us enter to our study, our work. So the plant species Agelanthus brenuis were harvested in the central region of Cameroon in the locality called Ngwaikele in 2018. So the plant was identified by a botanist in the Yaoundé where the voucher specimen is secure, and the plant were chopped and dry to yield 2.6 kilograms. And the powder were extracted using dichloromethane methanol 1,1 in the 78th hour to yield 206.5 grams of crude extract. So the crude extract was partitionated using liquid extraction to yield three main fractions: N-hexane fraction, ethyl acetate fraction, and N-butanol fraction. So the two less polar fractions, N-hexane and N-acetate, were subjected to column chromatography on silica gel and allow us to isolate 14 compounds. So we isolate using the column chromatography 14 compound, and the compound were characterized using NMR analysis 1D and 2D NMR and mass spectrometry. So this slide presents the structure of the compound isolated. So we have three pyrrole, among which the first one is new, not yet isolated and not yet synthesized. And the three other ones are one fatty acid and two phenolic acid. And the three last one are triterpenes. Also here in this slide, we have one steroid and four flavonoid isolated. So crude extract fraction and some isolated compound and standard drug gentamicin were evaluated for their antibacterial properties against 4 gram positive and gram negative bacterial species using p-iodo-nitrotetrazolium chloride microdeletion techniques. So this is the protocol of the antibacterial activity that we perform. So what we used was gentamicin obtained from Sigma aldris, DMSO MHB was used to dissolve the tested sample. The microbial growth indicator used was p-iodotetrazolium chloride. So, a four-strain bacterial species isolated from wastewater intended for human consumption were investigated in this work. So, they are the bacteria that we tested. So, Escherichia coli, Streptococcus faecalis, Salmonella typhi, and Vibrio cholera. So, in the next slide, we will focus on Salmonella typhi because Salmonella typhi is the bacteria responsible for typhoid. So those bacterial species were chosen because of their importance in hygiene and public health in indication of the microbiological quality of human pathogen germs. So determination of MIC and MBC of the sample. The mix of the sample were detected following the addition of 0.2 milligram per milliliter of p-iodotetrazolium chloride and incubation at 37 degrees C for a period of 18 to 24 hours. So the minimal bactericidal concentration of sample were determined following the addition of 150 microliter of culture brought into new plate. Then the volume was complete to 200 milliliter by adding a volume of 50 microliter of the wells contained with a concentration greater than or equal to the mix. Sorry. So the mix, the MBC MIC ratio were calculated and allow the characterization of the activity of the given antibiotic. So these are the results that we obtained. So the table 1 here presents the minimal inhibitory concentration in microgram per milliliter of the sample on the bacterial species tested. So this is so here the last line you have the MIC values of the reference drug positive control, and here you have the MIC in the table. You have the MIC value of the sample tested. The two first line are the result of the crude extract, and the second one are the result of the fraction, and the last ones are from 1 to 14 are compound isolated. So we can realize that all the tested products except compound 3 here show antibacterial activity with MIC ranging from 0.43 to 8.88 exposant 3 microgram per milliliter. Compound 14 and 9 show better activity on all the tested bacterial strain with MIC ranging from 0.43 to 27, 0.77 microgram per milliliter. So the minimal bactericidal concentration of the different extract, fraction, and isolated compound were determined, and the MBC/MIC ratio calculated. So this ratio varied between 1 and 10 for the bacterial species tested. So the obtained MBC results and the calculated ratio are present in the table 3 and 4. So the table here you have in table 2 and 3. Okay. Considering the MBC/MIC ratio, the product tested exert either bactericidal or a bacteriostatic effect on the bacterial species tested. Only the sample CLE, EAFL, and compound 14, 2 and 8, 2, 8 and 9 have a bactericidal effect on all the bacterial species tested. So now focusing on Salmonella typhi, which is a bacteria responsible for typhoid, we can say the new compound 1 exert bactericidal activity on the E. coli, yeah, and Salmonella typhi, which is the strain responsible for typhoid. So our new compound can be used in the next future as a target, as a lead compound to the discovery of new antibacterial or new anti-salmonella treatment. So, in conclusion, the goal of our study were to isolate and evaluate the antibacterial activity of Agelanthus bruneus. At the end of the study, 14 compounds were isolated among which 3 pyrrond with 1 new, 1 fatty acid and 2 phenolic acid, 3 tritapene, 1 steroid and 4 flavonoid. So all the test examples except compound 3 show anti-salmonella activity. Anti-salmonella because of typhoid. So at the end we focus on the typhoid. So anti-salmonella activities with compound 9 and 14 being the more active compared to gentamicin used as positive control. So all these results show that the traditional use of Agelanthus bruneus extract in reducing the flow of population bacteria in water and in the treatment of infectious diseases could be an alternative in the fight against typhoid. So in the next feature, you will work with biologists to assess
Hello, Dr.
the.
Thomas. Can you please check your slides again? We are unable to see your present slide.
It's okay?
May I know which slide you are presenting now? You are in the 12th slide here.
Okay. So,
Please change your. Please open your PPT and change your slide.
okay. Mm,
Can you? Okay.
Okay.
Okay, you can
Okay.
continue.
Okay. In conclusion, all the test examples except compound 3 show anti-salmonella activities, with compound 9 and 14 being the more active compared to gentamicin use as positive control. All these results show that the traditional use of Agelanthus bruneus extract in reducing the flow of polluting bacteria in water and in the treatment for infectious diseases could be an alternative in the fight against typhoid. So we are currently working with biologists to assess the cytotoxicity of this product and the new one more precisely. And we are also studying the polar function of the different parts of this plant, leaves, stem, and flowers. We are also looking for a collaboration to isolate the secondary metabolite from fungi because we have to preserve the biodiversity. And the method we are using now use a quantity of plant material. So I will not finish this presentation without thanking Professor Thomas Storr and Richard Stephenson from the School of Chemistry of University of East Anglia for the NMR analysis, and Professor Nobel Sevald for the Chemistry Department of the University of Bielefeld for the for the research grant for 6 months that I've spent there, and also Dr. Lucian Malis Mungan for the biological analysis of the samples. So thank you for your kind attention.
Thank you, Dr. Thomas, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Dr. Thomas, for your presence and presentation.
You're
Hello,
welcome.
Dr. Now let's move on to the next presentation. Hello, Dr. De Ferra. Are you ready with your presentation now? Hello, Dr. De Ferra. Can you please unmute yourself, Dr. De Ferra? I have made you as a presenter. Kindly click on the screen button to share your screen.
Yeah, okay. Can you hear me?
Yeah, you're audible. Kindly share your screen now.
Okay.
Yeah, your screen is visible too. Open your PPT presentation and start your presentation. And it was
Okay,
unshared
sorry, I've
again. Kindly
encountered
share it
an
again. Can you please share your screen again? Yeah. Don't close the GoToMeeting app. Just
Is
switch
it
to
visible?
the PPT mode. Yeah, it is visible.
Okay. Is it visible?
Yeah, it is visible. Can you
Hello?
open your PPT and...
Okay, thank you
Yes, it
very
is
much
visible. You can
for
start
your...
your presentation.
Okay, thank you very much for your invitation. Sorry for the disturbance because I've encountered a network connection problem. Saying this, I'm Ferrar Kitu from Lega University and lecturer of health economics, management and policy. Now, our presentation title which is entitled as Adverse Event Following Immunization of COVID-19 Vaccination among Healthcare Professionals in Ethiopia, which is an online survey that was conducted in our country. Can you hear me? Hello? Hello? Our presentation outline is almost this, and to start from the introduction, coronavirus disease 19 is an infectious disease caused by coronavirus or SARS-CoV-2, which was first reported in late December 2019 in Wuhan, China, and later it was assessed by WHO and declared as a public health emergency of international concern. And again, the disease was rapid globally and spread, and later it was declared as a global pandemic. When we come to our country, Ethiopia, it was first confirmed in March 13, 2020, which was reported by Federal Ministry of Health of Ethiopia. Nowadays, we are living in the pandemic situation, given its rapid spread and occurrence of new variants of COVID-19 vaccine, COVID-19s. Can you hear me? And as of 28 January 2020, globally, about 366 million cases and 5.6 million confirmed diseases were reported. And when it comes to our country, Ethiopia, more than half of a million cases was reported, and about 7,346 cases were diseases were reported in our setup. Saying this, the pandemic was exerting a negative impacts on different aspects, especially on health, social, economic, and the well-being of the people, which can result in the interruption of health system, which result in disease burden and economic burden. Can you hear me? To prevent this pandemic and understand related consequences, social and public health mitigation measures have been proposed and being implemented in the world. And additionally, researchers and pharmaceutical companies are collaborating to develop safe and effective vaccines of different types nowadays. And it is an effective way of combating the conditions. Particularly, it is an essential pillar for controlling the COVID-19 vaccine, COVID-19 disease. And nowadays, hundreds of the vaccine are on trials, and some of which have passed the acceptable efficacy and safety standards. And even almost greater than 9 vaccines have been reached emergency use lists. Based on this, Federal Ministry of Health of Ethiopia launched the vaccine to be given for the community in the 13th of March 2021. As a result of inadequate supply of the vaccine, different aspects, they have prioritized health high-risk groups, especially health workers, elderly, and chronic comorbidities, to receive the initial supply of the vaccine. And nowadays, about 61% of the world population has received at least 1 dose of the vaccine. And in the world, about 10 billion doses have been administered. And in low middle income country, only 10% of the people have received at least 1 dose. And in our setup, about over 10 million doses have been administered. Of these, 2.7 million of the people have been fully vaccinated. And although the vaccines have been administered for the different communities, the vaccine are not free from side effects. And importantly, some adverse effects of health problems following the vaccine may be due to the coincidences and are not caused by the vaccine. And also, false information as well mistrust can result in anxiety and in vaccine hesitancy for the community, which can result in hesitancy of toward receiving the vaccines. Also, manufacturers have included the potential post-vaccination side effects. Also, the post-market adverse effects and safety-related studies have been conducted across different countries. Of this, Afghanistan have reported about 93.5% of adverse effects following immunization, and next, the Korea have reported about 90% of the adverse effects following immunization, and all others are the reporters of adverse effects following immunization in the different aspects and from different worlds. To the level of our authors or to the level of our knowledge, there are no prior study based on adverse effects following immunization. And even though studies have been conducted in different worlds, they have focused on a specific vaccine, either full dose dosage, or view in different aspects and in different perspectives. So our intention is to fill this gap. So we have conducted our research paper on adverse effects following immunization of COVID-19 vaccination among healthcare professionals in the Ethiopian context. And we have an objective, that is a general objective, to assess the magnitude of adverse effects following immunization of COVID-19 vaccination. And it is factors. As a specific objective, we have determined adverse events following immunization of COVID-19 vaccination. And we have identified factors associated with COVID-19 vaccine. Also, we have a methodology parts. The study was conducted in Ethiopia from June 1st to 30, 2021, using cross-sectional study design. And all employed healthcare professionals working in health institution of Ethiopia were the source population, and all those selected healthcare professionals or surveyed healthcare professionals were the study population. In fact, here we have a study unit of analysis. Our unit of analysis is our participants, and we have included those participants whose age were greater than 18 years, as well as those participants who were residents of Ethiopian, or to mean that they have to be Ethiopian resident at this moment. and they have to understand English language, because in our setup we have different language to be given for different regions, and we have excluded those who couldn't access the internet, and incomplete reports were excluded. And when we come to the sample size, we haven't predetermined the sample size because it's a survey. However, we have reached about 522 healthcare professionals who were participated in the study within the study period. And the sampling technique, we have developed, or we have used a Google Form platform. That was the link that we have creates the questionnaires on that, and we have sent that questionnaire to this most important social media, especially on Facebook, emails, and Telegram, using convenience and snowball sampling techniques. And as a dependent variable, adverse event following immunization of COVID-19 were the dependent variables, and we have some variables that are independent variables, especially under social demographic and economic variables, professional work area related variables, health status and exposure related variables, vaccines, and adverse event related variables. So we have operationalized that adverse event following immunization in our in our case. It means that it is any untoward medical occurrence which follows immunization and which does not necessarily have causal relationship with the usage of vaccines. Based on this, we have done a regression analysis. So our data collection instrument was, as I have mentioned, this online platform, it was Google Form, and then sent to the authors network, and using semi-structured questionnaire, which was adapted by reviewing different literatures. When we come to the data quality assurance, we have conducted different data quality assurance at different stages before, during, and after data collection. Again, we have translated and retranslated our questionnaires, and then pre-tests in Wulaga University Referral Hospital. And in regard to data analysis, we have extracted from Google Form what we have collected from the participants, then exported to Excel, and later we have exported to statistical package for social science version 25. And we have received an ethical approval from Institutional Review Board of Wulaga University. And when we come to the results and the discussion parts, a total of 522 participants were involved in the survey, and majority of the participants were aged from 25 to 34 years, with a mean age of almost 31 years, with a standard deviation of almost 4.7. And the majority of our participants were male in sex, Oromo in ethnicity, Protestant in religion, and they are about 7% were married. When education-wise, almost greater than 80% were BSc and MSc holders, and majority of the participants were the urban residents and semi-urban residents. And when we come to the family size, almost 13.8% were have greater than or equal to 5 members in their households. And this is a table that I have before. So among 522 study participants who were involved in the participants, about 62% were vaccinated with the COVID-19 vaccine. At least one of these, 75% of them have encountered or have experienced adverse events following immunization. This finding was higher than study done in different countries, especially in Indonesia, in Togo, in South India, and in other comprehensive specialized hospitals. This might be due to sociocultural environment difference and difference in time period of considered for data collection, as well as type of vaccine considered. Because in our case, we have considered any type of vaccine, but the study might have considered either the full dosage as well as specific vaccines. This finding was lower than study done in Afghanistan, in which the study reported about 93%, and in Korea, it's reported as 90%. The probable reason might be due to the study population different in that area, because most of the study what we have compared with that is the medical students. But in our case, their employed healthcare professional experienced, and the study finding was in line with the study done in United Kingdom, which was reported as almost 72%, and low middle income countries. Major adverse event following immunization was reported in this finding, and the major adverse event following immunization report was to lesser or greater extent comparable with different studies across the globe, as well as vaccine safety manual prepared by World Health Organization. In this case, almost majority of the adverse event following immunization was reported as injection site pain, redness, and tenderness, and some of the adverse events following immunization reported were systemic reactions. Some of the common adverse events reported was higher than the study done in Nova Scotia as well as Indonesia, in which the report indicates lower than our study finding, but higher than study finding. Sorry, and in other case, the study finding was lower than the study conducted in Korea and in Togo. This might be due to the current study focused on any of the vaccines than specific vaccines, because that study have focused on specific vaccines, and also in line with the study conducted in Afghanistan, in which the study indicates 68.3%, as well as low-income countries, which indicates about almost 75.8%. Regarding to perceived severity, about almost 59.3%, 34.6%, and 6% were mild, moderate, and severe infection, respectively. Most severity of the report have reported adverse events was mild to moderate reactions. This finding was comparable with another study done in different aspects and in different countries, especially in Afghanistan, in Korea, and Southeast Ethiopia. However, severity of adverse event following immunization reported in this study was higher than in the study conducted in Minas Gerais, which was reported as 3%, but in our case, it was about 6%. In Korea and South India, and all these are the discrepancy that have been observed in our case and in that country. This discrepancy might be due to heterogeneity in the population the study have considered, and some of the studies were institutional based, whereas severity of adverse events following immunization indicated was lower than in Nova Scotia and in Togo, in which 17.7% were reported in Nova Scotia, as well as 23.8% was reported in Togo. This might be due to difference in types of vaccines over which the severity was assessed. Additionally, studies conducted in Ontario, as well as in British Columbia, and also low-income country, showed a similarity in severity, in which the severity reported was almost 6%, 7%, and 6% respectively. Additionally, we have seen an association between the adverse event following immunization and the factors in relation to that. In fact, we have entered in spaces, and almost 15 variables were candidate for this study, and of this candidate variable for the multivariable logistic regression analysis, finally in the multivariable logistic regression, almost 5 of them were significantly associated with adverse event following immunization. Of this, marital status was 1. In this case, married respondents were 4 times more likely to encounter adverse events than a single respondents, which was supported by Togo in either comprehensive specialized hospital and southern Ethiopia, which might be due to similarity in population characteristics and even in the similarity in health system. And also married individuals might have high risk perception. The other thing is family size. When we come to the family size, those participants whose their family were greater than or equal to 5 per households were 5 times more likely to encounter adverse events than their counterparts. And another, the family tested for COVID-19 was another significant variable that we have obtained. But in case those participants who their family tested for not tested for COVID-19 were 61% less likely to encounter adverse event following immunization than those of its counterparts, which was supported by the study conducted in Ghana, in which 84% of the household members were not diagnosed for COVID-19. Another thing is the family support to take the vaccine. In this case, those respondents who their family support to take, who has no family support to take vaccine were almost 3.5 times more likely to encounter adverse effects, adverse event following immunization than the other parts. This was supported by a study conducted in low-income country. The probability might be due to fear of sign and symptom associated with misinformation. And heard anything about COVID-19 vaccine adverse event is another significant variable what we have obtained. In this case, almost those participants who have heard anything bad about the COVID-19 vaccine adverse events were 4 times more likely to encounter adverse event following immunization than the counterparts, which was supported by study conducted in Ghana and low-income country. And the very important concept here is the concern about the vaccine could cause adverse effects. In this case, those respondents who very concerned about adverse event the vaccine could cause adverse event following immunization were 6 times more likely to encounter adverse event following immunization than not at all concerned. This finding was in line with study conducted in Ghana, in Togo, as well as in low and middle income countries and Amara regions of Ethiopia. The concern about side effects are the most common reason reported in this country. That is our probable justification. And in this study, we have this study has its own limitation. The first one is the study was conducted only after the first dose was administered in our case, and probably we have missed the second dose that have been administered. And the other thing is we have used non-probability sampling techniques because it was, at the time of lockdown, it is impossible to conduct face-to-face interview, as it is somewhat difficult to generalize to the general population, as well as healthcare professionals who have no access to the internet were not participating in this case. This might underestimate our finding, and it is an online survey. We might probably face a low non-response rate, and the study was a cross-sectional nature, as it is somewhat difficult to show the cause and effects. When we come to the conclusion and recommendation, as a conclusion, almost 3/4 of the study participant has experienced adverse event following immunization. However, adverse events following immunization experienced was to lesser or greater extent comparable with different studies across different world and different countries, and to the vaccine surveillance monitoring, which was prepared by WHO and manufacturing company has reported. Common symptoms were injection sites and majorly range from mild to moderate, and another thing is the factors associated with adverse events following immunization. Almost I have discussed with in the discussion part: marital status, family size, tested, family support, and heard anything bad about the vaccine, as well as concern about adverse event following immunization were the factors that were associated with this adverse event following immunization. To conclude this, health managers at different levels, whether in our country as well as in different world, should have to encourage full vaccination to any healthcare professionals, and even they have to use as a baseline information as a policy implication, and also for the vaccine to the general population that have to be given for our community. In fact, no time we are giving to our community for age greater than 12 years. So this research will be an input for the policy designers, policy implementers,
I think there is a network connectivity issue from Dr. DeFera end. We'll wait for a few minutes and we can for Dr. DeFera. I hope Dr. DeFera has still persist network issues. So by this, we come to an end of day two webinar. I would like to thank all the attendees and participants of today's web event. Hope you enjoyed the conference and gain knowledge from the sessions, and we'll be glad if you could join us in the next in-person conference, which is scheduled in November 14-15 in Dubai 2022. And the dates will be informed to you by mail. We will be
Hello.
Hello, Dr. DeFera, are you ready to conclude the meeting?
Okay, thank you very much. I think I've concluded that. Can it be heard?
Okay, Dr. DeFera, thank you for your presentation. By
Hello.
this, we are going to end the meeting today now.
Okay, thank you. If you have if any question, I'll react on that.
Okay. Okay, and now we'll be glad if you could join us in the next in-person event conference, which is scheduled in November 14-15, 2022 in Dubai, and the dates will we will be very happy if you could share your opinion and feedback with us. If you have any queries, you can mail to us. We'll resolve. Thank you.
Okay, thank you.